In 1984, this Australian doctor named Barry Marshall was just so completely fed up with his
Oh,
colleagues.
yeah, they were entirely ignoring his medical data.
Right. So to prove his point, he took matters into his own hands. He literally brewed up this cloudy broth in a petri dish a broth swarming with billions of live bacteria, and he just drank it.
He downed it like a shot, which is, incredibly reckless.
Totally reckless. But today we are diving into the invisible world that drove him to do that. Welcome to the deep dive. We are exploring this ultimate hidden empire, this bustling, frankly bizarre metropolis that exists entirely right under our noses, on our skin, and inside our stomachs, but is completely invisible to you.
Yeah, and it's an empire that dictates human health, it drives the global environment, and it honestly constantly challenges our very definition of what even constitutes a living thing.
It really does.
And for this deep dive, we are drawing on a really fascinating stack of materials for you today. We've got historical accounts from the very first people who peered through crude lenses, the evolving biological definitions of what a microbe is, and these wild case studies that just shatter conventional medical wisdom.
Oh, yeah. We're talking about scientists drinking their own experiments, organisms the size of a human eyelash, and the ongoing debate over the boundary of biology itself.
It's a lot of ground to cover.
Okay, let's unpack this. Our mission today is to answer a deceptively simple question. What is a microorganism? We want to uncover the kinds of microbes that rule our world and explain why they matter so much to our health, our diseases, and the environment.
Right.
But before we can even talk about how these invisible entities control our world, we have to actually define what they are, which it turns out is a lot harder than it sounds. So let's start with the baseline. What is the physical threshold we are talking about here?
Well, the standard working threshold for a microorganism, or a microbe, is basically anything too small to be seen with the unaided human eye.
Okay.
So for you and me, the physical limit of our visual acuity is about 0.1 millimeters.
That is tiny.
Very tiny. So to actually see these entities, you typically need magnification of at least 10 to 1,000 times.
Yeah.
And under that broad umbrella, you have bacteria, archaea, protists like amoebae, and microscopic fungi.
Like yeasts and molds, right?
Exactly.
I mean, that sounds like a really neat, tidy categorization. But biological classifications are kind of notorious for being overturned. I imagine that, for a long time, early scientists just looked through a lens and threw everything that looked like a squiggly dot into one giant bin labeled bacteria.
Oh, they absolutely did for a very long time. Actually, until the late 1970s, microorganism was largely treated as a synonym for bacterium in a lot of contexts.
Wow.
But then Carl Lewis, who was a microbiologist at the University of Illinois, he completely changed that paradigm in 1977.
Okay, how so?
Well, instead of categorizing microbes by their shape or how they behaved in a petri dish, which is incredibly misleading, because a sphere is just a sphere, Woese looked at their genetic blueprints. Specifically, he sequenced their ribosomal RNA.
Okay, let me make sure I understand the mechanics of why that matters. Ribosomal RNA isn't just like random genetic material, right? It's the core machinery a cell uses to build proteins.
Yes, exactly.
So comparing RNA instead of just looking through a microscope is kind of like ignoring the outer casing of a smartphone and looking directly at the underlying operating system code.
That is a perfect way to look at it,
I know.
because that core machinery, it evolves incredibly slowly over billions of years, so it acts like a molecular clock.
Oh, that makes sense.
And by comparing those underlying RNA sequences, Woese discovered that a group of microbes everyone just assumed were odd bacteria were actually something else entirely.
Wait, really?
Yeah, these were extremophiles living in boiling hot springs and super salty lakes. And structurally, their genetic code was as different from bacteria as bacteria are from humans.
So they looked the same on the outside, but their internal software was completely alien.
Precisely. Which is why Woese proposed an entirely new domain of life for them, called archaea.
Yeah.
And this gave us the standard three-domain system we use today. You have bacteria, archaea, and eukarya, which includes us, right along with plants and animals.
But I'm guessing the establishment didn't just accept that overnight.
Not at all.
Yeah.
Senior biologists were intensely skeptical at first. I mean, they completely dismissed his genetic tree. They preferred the old visual classifications. It took a decade of intense debate before archaea was widely accepted.
Science really does love to hold on to its dogmas. But speaking of things that completely mess with the definition of life, we have to talk about viruses.
Oh, yes.
Because whether a virus even counts as a microorganism seems to trigger massive arguments among biologists.
It really does. The debate centers around the classical definition of life. To be a living cell, an organism needs a membrane to contain itself, it needs to metabolize energy to survive, and it has to be able to reproduce independently. Viruses fail every single one of those tests. Basically, a virus is just an inert package of genetic material, DNA or RNA, wrapped up in a protein code. Outside of a host cell, it has no metabolism. It literally does absolutely nothing.
It's not even alive. It's like, okay, it's like a terrible burglar who doesn't know how to cook.
A burglar.
Yeah, think about it. He doesn't know how to cook, so he breaks into a restaurant kitchen, right? He doesn't bring any food, no pans, nothing. He just forces the chefs at gunpoint to cook his own family recipe over and over and over again.
That captures the physical mechanism beautifully.
Thank you.
The virus provides the recipe, the malicious code, but the host cell provides all the energy and machinery, like the chefs, to build the copies. And because of this, many microbiologists insist viruses are just biological entities, not actual organisms.
But people still call them microbes anyway.
Right. Virologists and the general public often lump them into the microorganism category anyway, simply because they're microscopic, they cause disease, and they evolve.
And just to complicate things even further, the sources mention giant viruses, which sounds like a total contradiction.
Oh, it really blurs the lines. In 2003, researchers discovered virus, which is about 750 nanometers across. That is so massive, it was initially mistaken for a bacterium.
Wow.
And then a decade later, they found Pandora viruses. These carry genomes larger than some actual living bacteria, and they even possess genes for protein synthesis.
Wait, which is machinery a virus supposedly never needs, right? Because they just use the host.
Exactly. These exceptions blur the biological lines so severely that some scientists argue we need a fourth domain of life just to categorize them.
Okay, but let me push back on this a bit. If the fundamental definition of a microbe is that it's invisible, smaller than that 0.1 millimeter limit you mentioned, then what happens when a microbe physically breaks the rules?
What do you mean?
Like, what if something is biologically categorized as a bacterium, but I don't need a microscope to see it?
What's fascinating here is a discovery made as recently as 2022. Researchers looking at decaying mangrove leaves in the Caribbean found a bacterium called Theomargarita magnifica. And it grows into visible, thread-like filaments up to 2 centimeters long.
2 centimeters, that is the size of a human eyelash.
Yes.
That is not micro. That is something you can literally pick up with tweezers.
It's thousands of times larger than typical bacteria. And finding it completely shattered a biological assumption that has stood since the 1880s.
What assumption?
Well, for over a century, the consensus was that there was a hard physical ceiling on bacterial size, because bacterial cells don't have complex internal transport systems like our human cells do. They rely on simple diffusion.
Which is basically like trying to deliver mail in a massive city without any roads or delivery trucks. You just toss the packages into the air and wait for the wind to randomly blow them to the correct address.
Exactly the problem. The larger the cell gets, the longer that random diffusion takes. So the assumption was that if a bacterium grew too large, diffusion would become impossibly slow, the internal chemistry would just stall out, and the cell would die.
So how does an eyelash-sized bacterium cheat physics?
It circumvents the diffusion problem entirely. Instead of keeping its DNA in one central clump and waiting for instructions to float across a massive space, Theomargarita magnifica packs its DNA into thousands of little membrane-bound sacs.
Oh, wow.
Yeah, they're distributed all throughout its enormous body. So no part of the giant cell is ever far from its own local copy of the genetic instructions. It completely decentralized its own operations.
That is incredible. Nature clearly does not care about our neat textbooks. It just finds a mechanical workaround.
Truly.
But, this whole struggle to categorize the invisible world, it's really just the latest chapter in a very long story. I mean, it took humanity centuries to even admit these things existed in the first place.
It did. The shift in human perspective took immense effort. The entire field really originates in the 1670s with Anthony van Leeuwenhoek.
He was a Dutch draper, right? Like a fabric merchant, not a formally trained scholar.
Exactly. But he was grinding his own lenses to inspect the thread count of his fabrics. And he got incredibly good at it.
Better than the actual scientists of his day.
Much better. While other early scientists were using compound microscopes, which, stacked multiple lenses but suffered from terrible blurring and distortion, Leeuwenhoek perfected the art of creating tiny, perfectly spherical, single-glass lenses.
So it was just one simple lens?
Yes, his simple microscopes achieved 200 to 300 times magnification with stunning clarity. By 1674, he was writing to the Royal Society of London about what he called animalcules swimming in pond water.
The detail he achieved is just astonishing. I mean, the sources note that in 1683, he scraped the white plaque off his immune teeth, put it under his lens, and sketched distinct shapes of what were undeniably living bacteria.
Yeah, he documented the microscopic forest living inside his own mouth.
Which is gross, but amazing. Yet this was 170 years before anyone seriously linked those squiggly shapes to human disease.
Right. Leeuwenhoek was driven by sheer curiosity. The concept that these tiny entities could invade a human body and cause illness just didn't exist.
Yeah.
In fact, the Royal Society was so dubious of his initial claims that they had to send an independent team to Delft just to verify he hadn't fabricated the whole thing.
Which really sets the stage for one of the most tragic blind spots in medical history. Because once humanity knew these things existed, you'd assume we would quickly realize they were making us sick.
You would think so.
But instead, we get the story of Ignaz Semmelweis in 1847 Vienna.
Oh, the tragedy of Semmelweis. He was a Hungarian physician running a maternity ward, and he noticed a horrifying statistical anomaly. Women in the ward attended by medical doctors were dying of childbed fever at a vastly higher rate than women in the adjacent ward run entirely by midwives.
The critical variable being that the doctors were splitting their shifts. They were coming directly from performing autopsies on corpses to delivering babies.
Yes. Semmelweis deduced that the doctors were physically transferring something from the corpses to the mothers. He called them cadaverous particles.
Right, because germs weren't really a concept.
Exactly. But based purely on this observation, he mandated that all doctors scrub their hands with a strong chlorinated lime solution before touching a patient.
And it worked, right?
The mortality rate immediately plummeted.
So he cracks the code, saves lives, and is instantly hailed as a medical pioneer.
Sadly, no. He was ridiculed and pushed out of his profession.
Wow.
The medical establishment rejected his findings entirely because they lacked a theoretical framework for invisible disease agents. The prevailing dogma at the time was miasma theory, which claimed diseases were caused by bad air or spontaneous imbalances in the body.
So they just refused to believe it was something physical on their hands.
Yes. And beyond that, doctors consider themselves gentlemen. The implication that a gentleman's hands could be dirty, that the doctors themselves were the vectors of death, was treated as a personal insult. Semmelweis was discredited and, tragically, ultimately died in an asylum in 1865.
The bitter irony there is that in the 1860s, while Semmelweis was dying in disgrace, Louis Pasteur in France was finally proving the mechanical reality of germs.
With his famous swan-neck flask experiments.
Right. He takes a glass flask filled with nutrient broth, boils it to kill everything inside, but then he heats the glass neck and stretches it out into a long S-shaped curve.
The brilliance of that curve is that it leaves the flask open to the air, which satisfied the critics who said air was required for life to spontaneously generate.
But the physical curve acts as a gravity trap.
Exactly. Airborne dust and microbes settle in the bottom of the bin and they can't travel up and into the broth. As long as the curve remained intact, the broth stayed completely sterile.
It proved that life doesn't just magically spring from non-living matter. It has to be introduced physically, which leads directly to Robert Koch in Germany, who takes Pasteur's concept and weaponizes it into a really rigorous methodology.
Yes, Koch was working on anthrax and tuberculosis in the 1870s and 80s, and he establishes the germ theory of disease by outlining a set of criteria in 1884. We know them as Koch's postulates.
It's essentially a biological chain of custody, like a rigorous detective's checklist for pinning a crime on a suspect, right?
This is a great analogy.
Like step one, you have to find the specific microbe present at every single crime scene, so in every organism suffering from the disease, but absent in healthy ones.
Right, and then step two, you must extract that suspect and bring it in for questioning. You have to grow that microbe outside the body in a pure culture.
Okay, and step three, you introduce that cultured microbe into a completely healthy host, and it must mechanistically trigger the exact same crime, the exact same disease.
And finally, step four, you have to extract the microbe from that newly sick host and prove it's identical to the original culture. You catch it red-handed again.
Right.
If we connect this to the bigger picture, this methodology, which was developed by Koch, Pasteur, and a botanist named Ferdinand Kohn, it fundamentally rewired human knowledge.
How so?
They transformed microbiology from Leeuwenhoek's hobby of looking at things under a lens into a predictive, rigorous science. They provided the framework to actually prove disease causation.
Now, you would think the medical establishment would learn from the tragedy of Semmelweis and the rigors of Koch, right? You'd think they would learn not to blindly dismiss crazy-sounding microbial theories.
You would hope so.
But fast forward 100 years to the 1980s, and they were still making the exact same mistakes.
Oh, absolutely. The deeply entrenched dogma of the 20th century dictated that stomach ulcers were a lifestyle disease. Doctors believed they were caused by stress, spicy foods, and excess stomach acid.
Because the biological assumption was that the human stomach is a vat of acid so harsh that it's functionally sterile, they assumed nothing could survive there.
Right. But Barry Marshall and Robin Warren, looking at biopsy samples, kept finding this spiral-shaped bacterium, later named Helicobacter pylori, thriving right in the inflamed stomach linings of ulcer patients.
But the medical community rejected their findings, right? They just said it was a secondary infection.
Yes, they argued the stress caused the ulcer and the bacteria just showed up afterward. And Marshall and Warren couldn't fulfill Koch's postulates because they didn't have an animal model. Lab mice just didn't develop the ulcers the way humans did.
And here's where it gets really interesting. Marshall decides that if the establishment won't believe the data, he has to become the host himself. Which brings us back to our intro, him downing that Petri dish in 1984.
He drinks a cloudy, swarming culture of H. pylori.
Unbelievable.
And within days, he develops acute gastritis. He then performs an endoscopy on his own stomach to biopsy the inflamed tissue, proving the bacteria had colonized his stomach and caused the damage.
And then he just cures it with antibiotics.
Exactly. Finally, he takes a simple course of antibiotics and completely cures himself.
So he weaponized Koch's postulates on his own digestive tract.
He did, and they eventually won the Nobel Prize for this in 2005.
Which is amazing. But this story forces a really uncomfortable question about modern medicine. I mean, if doctors were so aggressively wrong about ulcers just a few decades ago, what other textbook facts about our bodies are actually just unverified assumptions?
It's a fair question.
Take the most famous microbiology statistic of all, the one everyone repeats. The idea that we are outnumbered by our own bacteria 10 to 1. Is that even real?
That statistic is a perfect example of modern dogma. For decades, scientific papers, textbooks, and documentaries repeated that exact metric, 10 bacterial cells for every one human cell.
Right.
But in 2016, a research team at the Weizmann Institute in Israel, led by Sender, Fuchs, and Milo, decided to actually audit the math.
I am guessing they found out the number was completely fabricated.
Well, not fabricated, but it was basically an academic urban legend. They traced the 10 to 1 claim back to a single, unverified estimate published in 1972.
Wait, really? Just one paper?
Just one guess, really. It was based on the assumption that the entire digestive tract was as densely packed with bacteria as human feces. When the Weizmann team actually ran the real numbers, calculating the distinct volumes of different organs and the actual density of microbial life, the ratio completely collapsed.
So what is the actual ratio?
It is remarkably close to 1 to 1. For a reference adult male, the estimate is roughly 38 trillion bacterial cells to 30 trillion human cells.
Okay, so it's practically a tie.
Yes. And the vast majority of those 38 trillion microbes are packed almost exclusively into the colon. The lesson here is that even precise-sounding science numbers can just be unverified urban legends.
But even at a 1-to-1 ratio, that still means 38 trillion microbes are hitching a ride on our biology. We are deeply physically entangled with these organisms on a massive scale.
We really are.
Which leads perfectly into why understanding their behavior, how they hurt us and help us, is an urgent matter of planetary survival. But I want to clear the record for you, the listener, right now. Are all these germs actually out to get us?
Oh, no, not at all. That is the most persistent misconception out there. We equate microbe with pathogen, but pathogens, the microbes that actually cause disease, are the extreme minority.
Really?
Yes. The vast majority of our 38 trillion companions are commensals, which simply coexist with us, or they're mutualists, which actively keep us alive.
Like gut bacteria digesting our food.
Exactly. They break down complex carbohydrates that human enzymes can't process, they synthesize essential vitamins, and they physically crowd out dangerous bacteria.
Yeah.
And on a planetary scale, soil bacteria fix atmospheric nitrogen, converting it into a form plants can actually use.
So without them, the global food chain just collapses.
Completely. They are the invisible scaffolding of the world.
But when we do encounter the dangerous ones, we have to understand the mechanics of our weapons against them. Which brings me to a crucial point for your everyday life, the biological distinction between viruses and bacteria. Why exactly won't your doctor give you antibiotics for a viral cold?
because of the structural differences we discussed earlier. An antibiotic is a chemical weapon designed to target specific machinery inside a living bacterial cell.
Right.
Some antibiotics physically dissolve the bacterial cell wall, while others jam the specific ribosomes the bacterium uses to make proteins. But remember, a virus isn't a cell.
Right. It doesn't have a cell wall.
Exactly. It doesn't have its own ribosomes either.
It's the terrible burglar. So pouring an antibiotic on a viral infection is like trying to fix a corrupted software file by smashing your computer screen with a hammer. It just completely misses the mechanism of the threat.
That's a perfect way to put it. And this raises an important question about the consequences of misunderstanding these microbes. By overusing antibiotics, prescribing them for viral colds, or dumping them by the ton into agricultural animal feed, we have applied massive evolutionary pressure on bacteria.
We're training them.
Yes, we are actively breeding antimicrobial resistance, or AMR.
Which is how we end up with things like MRSA, a staph infection that has evolved physical workarounds to survive the very drugs that used to eradicate it.
Exactly. The World Health Organization actually classifies AMR as a top global public health threat. And we also face the constant threat of zoonosis, which is the physical jump of a pathogen from an animal host into human populations.
Like the COVID-19 pandemic.
Yes, SARS-CoV-2 is a prime example of how a microscopic entity can literally reshape global society in a matter of months. And furthermore, we face bacterial threats like C. difficile or Bacillus anthracis, which causes anthrax because they can form endospores.
Now, what exactly is an endospore? Because it sounds like a sci-fi defense mechanism.
It is essentially a microscopic panic room.
A panic room?
Yeah. When the environment becomes hostile, the bacterium strips away all its non-essential material, packs its DNA into a dense, heavily armored shell, and shuts down its metabolism entirely.
So just play dead.
Basically.
Yeah.
And these dormant endospores can survive boiling water, harsh chemical disinfectants, and they can wait for decades in the soil until conditions improve.
Okay, that is deeply unsettling. But to balance the scales a bit, the microbial world also contains organisms with superpowers that could actually save us, right?
Absolutely.
Like even just a standard bacterium like Escherichia coli. Because it uses simple division, under ideal conditions, it can double its population every 20 minutes. The exponential math there means a single cell can produce billions of descendants in just a single day.
True. But if you want to talk about true superheroes, you have to look at Deinococcus radiodurans.
Oh, Conan the bacterium.
Exactly. This bacterium can survive radiation doses thousands of times greater than what would be lethal to a human being.
Wait, how does that mechanically work? Because radiation doesn't just poison you, it physically shatters the DNA ladder into pieces. If a human's genetic blueprint is shattered, our cells can't function and we die. How does the bacterium survive its blueprint being shredded?
It actually anticipates the damage. Deinococcus radiodurans keeps 4 to 10 backup copies of its entire genome at all times.
No way.
Yes, when radiation shatters its DNA, the bacterium activates specialized protein rings that grab the thousands of fragments, line them up against the backup copies, and flawlessly stitch the entire genome back together within hours.
It just reassembles its own shattered blueprint.
It does. And scientists are studying its rapidly repairing mechanisms right now to understand radiation resistance and perhaps apply those traits to sterilizing equipment for deep space travel.
Wow. And they aren't just useful for space either. The sources point out that when the Exxon Valdez and Deepwater Horizon oil spills happened, cleanup crews relied on naturally occurring oil-degrading bacteria.
Right. Bioremediation.
Yeah. These microbes use specific enzymes to physically break down the complex hydrocarbons in crude oil and use it for energy. We are literally using them to digest our own environmental disasters.
It really highlights the dual nature of this invisible world.
Yeah.
I mean, they can be devastating pathogens, but they're also nature's ultimate survivors and environmental saviors.
So what does this all mean? If we pull back from the microscope, we realize that microorganisms are not just germs waiting to be scrubbed away. They are this wildly diverse, bizarre collection of entities. We've got giant bacteria with membrane sacs, inert viral code injectors, extremophiles stitching their own shattered DNA back together. They run the operating system of our bodies and the operating system of the planet.
We are just massive, walking ecosystems navigating their world.
Exactly. And as we wrap up, I want to leave you with one final kind of mind-bending thought about just how little of this world we actually understand, because the sources mention something called the Great Plate Count Anomaly.
Ah, yes. The Great Plate Count Anomaly. Despite everything humanity has learned, from Leeuwenhoek's single lenses to modern RNA sequencing, When microbiologists scoop up a sample of soil or seawater and try to culture those microbes in a laboratory, only about 1% of them will actually grow.
Only 1%.
Yes, the vast majority, 99% of microbial diversity on Earth, is known only through uncultured DNA. They refuse to grow in our labs.
So we basically know nothing about them.
Pretty much. They require complex, interwoven environmental conditions that we simply do not know how to replicate yet.
So I want you to think about that. If we already have Conan the bacterium and eyelash-sized microbes in the 1% we can study, what on earth is hiding in the 99% we can't? The next time you touch your skin or take a breath or feel your stomach rumble, remember, you are carrying an invisible universe. We'll see you next time on The Deep Dive.